Hello everyone, we are in our lecture series of overview and integration of cellular metabolism. We are continuing with individual amino acid metabolism and their disorders, and in today's class we will be discussing metabolism of glycine. Very important, we will be covering the chemistry of glycine, we will be discussing the overview of glycine metabolism, how glycine is synthesized, how it is degraded, what are the specialized products from glycine and the various disorders of glycine metabolism. So, it is a huge daunting task. So, buckle up, gather your own notebooks and we will try to cover the entire topic in this class, alright. So, glycine, as you all know, it is a very simple amino acid with only hydrogen in place of the carbon skeleton. It is a non-essential amino acid, means it can be synthesized in our body. Metabolically it is glucogenic amino acid, ultimately it will enter into the TCA cycle by any means possible, right. It is involved in 1 carbon metabolism, it is present in interior of many structural protein, for example collagen, every third amino acid is glycine, MCQ question, and multiple specialized products are produced from glycine such as hemes, purines, creatinine, etc. We will be discussing it all. So, this is the overview of glycine metabolism, which might seem very daunting to start with because there are so many things, right. But as we progress, we will again revisit the slide at the end of this class, and where we will see, we will tick off every possibility that has been shown over here. Now, first starting with the synthesis of glycine, it is done from 4 sources, alright. Serine, threonine, carbon dioxide and ammonia, that is directly by capturing of carbon dioxide, 1 carbon, and glyoxylate. So, let us see. From serine it is converted to glycine by the enzyme serine hydroxymethyl transferase. Serine is another amino acid, it acts as a source to produce glycine. Now, what happens? The beta carbon is removed, alright, the CH2H is removed and it is donated to this tetrahydrofolic acid, and this enters the 1 carbon pool. So, thus by this step glycine is actually participating in 1 carbon metabolism. Mind you, this reaction is also reversible, okay, it goes both ways. So, glycine can also be converted to serine. So, exchange of 1 carbon thus happens, and by doing so the alpha carbon of serine becomes the alpha carbon of glycine, alright. Next source is production from threonine. Threonine is also another amino acid, it is cleaved by the enzyme threonine aldolase and it gives rise to glycine and acetaldehyde, alright. Threonine aldolase, very important MCQ. Third source, that is production from carbon dioxide and ammonia, right, and another 1 carbon unit. So, how it is done? It is done, so the 1 carbon unit again it is donated by N5N10 methylene tetrahydrofolic acid, it is converted to tetrahydrofolic acid. The whole enzyme system is actually glycine synthase complex, alright, and it directly converts carbon dioxide and ammonia to glycine, alright. This is NADH dependent. So, NADH plus H plus is converted to NAD plus, it is a multi enzyme complex. This is also reversible, and the reverse process is known as glycine cleavage system, which we will be discussing very soon, alright. So, this is the third source of glycine production, and the fourth source is from glyoxylate. How it is produced from glyoxylate? By the simple process of transamination. So, glyoxylate along with alanine is acted upon by a transaminase enzyme or aminotransferase enzyme to produce pyruvate and glycine. However, this reaction strongly favors synthesis of glycine. It means whenever glyoxylate is present in body, it tends to form glycine. You need to remember this step and also the previous steps, because how glycine is synthesized and degraded, because any problem in this will lead to diseases, right. So, now we have discussed the synthesis of glycine from all the 4 ingredients, that is serine, threonine, carbon dioxide, ammonia as well as glyoxylate. Now, we will be discussing the degradation of glycine, how glycine is degraded, and the main mechanism of glycine degradation is known as glycine cleavage system, which is a very complex thing as it appears because there is so much thing written in the slide. It consists of 4 proteins as a part of the multi-enzyme complex, P protein, T protein, L protein and H protein, and it actually looks like this. This is the major route of glycine breakdown for animals. So, let us, it is actually very easy. So, let us follow along. The P protein is actually glycine decarboxylase, which again has got pyridoxal phosphate as a cofactor, which catalyzes removal of carbon dioxide from glycine. So, this is the P protein with PLP, it removes carbon dioxide from glycine, decarboxylation, right. The T protein, this is basically a transfer of aminomethyl group. T protein containing lipoamide, this actually releases ammonia. So, aminomethyl transferase, if you look at the T protein, it is transferring the aminomethyl group. So, here the aminomethyl transferase is occurring and ammonia is being released. Next, L protein is basically a dehydrogenase, it is similar to dihydrolipoyl dehydrogenase of PDH complex. Okay. This regenerates lipoamide. So, this is an FAD containing coenzyme, it actually regenerates lipoamide, and in controlling all of them, in connection to all of them is basically the H protein. Basically, it is a protein that is modified with lipoic acid, which interacts with all the other components of the cycle, which includes reductive methylamination catalyzed by P protein, methylamine transfer as well as electron transfer, that is reduction. So, all of this occurs in conjunction to break down glycine. Now, if we are looking into 3D structure, it might seem very daunting, but we can actually express them in a much simpler way. So, this is how it happens. So, first glycine decarboxylase acts, CO2 is given out, and ultimately all the other cofactors tetrahydrofolate, lipoamide, NAD, FAD, they are PLP, they are all acting to regenerate the intermediate, so that this cyclical reaction can take place. Basically, what we are dealing, we are reversing the glycine synthase complex. So, it means glycine is broken down into carbon dioxide, ammonia, and 1 carbon metabolism is happening, I mean 1 carbon exchange is happening. So, this is the most important route of glycine breakdown, alright. Next, we are discussing about the fate of glycine when it enter, how it can enter the glucogenic pathway. So, how it can enter the glucogenic pathway? See, glycine, as we already discussed, can be converted to serine by the action of serine hydroxymethyl transferase, SHMT, right. Thereafter, serine is acting as a precursor for glucose. How? Serine is acted upon by dehydratase enzyme, one water molecule breaks, goes out, it forms an amino acid, and ultimately one water molecule is again entering and ammonia is being removed. So, by deamination of the amino acid, it is forming pyruvate. So, glycine is being ultimately via serine, it is converted to pyruvate, right. So, now we are dealing with the specialized product from glycine. Okay. So, specialized products from glycine are many, and we will also be discussing the role of glycine along with them, right. So, they are creatinine, creatine phosphate and creatinine, heme, purine nucleotides, glutathione, how glycine is acting as a conjugating agent, role of glycine in protein, neurotransmitter. So, let us discuss it one by one. Okay. So, first the biosynthesis of creatinine. Creatinine synthesis starts from synthesis of creatine. Okay. So, creatine is basically a high energy compound that is synthesized and it is present in the muscle, and it is synthesized from 3 amino acid, glycine, arginine and methionine, very important. So, this is an MCQ. So, any other amino acid if it is mentioned, it can be a question. So, all of this are constituent of creatine except, so be mindful, right. So, let us see how it happens. The first step occurs in mitochondria of kidney and pancreas. It involves transfer of guanidino group from arginine to glycine catalyzed by an amidotransferase enzyme to produce guanidino acetate. So, this is what is happening in the first step. This group is being shifted to glycine and ultimately guanidino acetate is formed. Okay. This is the first step, amido transferase, amido group is being transferred, right. In the next step what happens, this guanidino acetate will be methylated. Who is the most common methyl donor in all metabolic reaction? That is activated methionine in the form of S-adenosyl methionine. Okay. S-adenosyl methionine ultimately methylates this guanidino acetate to form creatine, and this methylation occurs in liver. So, there is a simple methylation. So, addition of one methyl group that is happening over here. Okay. We got creatine. Now, in the next step what happens, this creatine is phosphorylated. Whenever we need phosphorylation, kinase enzyme will work. So, creatine kinase is the enzyme that phosphorylates this creatinine to produce creatine phosphate or phosphocreatine. This phosphocreatine is actually a ready energy source for muscle. So, let us deviate a bit, that this is the step 3, creatine kinase, alright. So, what happens, this creatine phosphate, we need creatine phosphate to go ahead to form creatinine, right, but this creatine phosphate when required can actually be broken down to creatine. You see, creatine is requiring an ATP to form creatine phosphate. So, this is a high energy compound. So, whenever the muscle needs ready ATP, creatine phosphate is broken down, alright. So, this reaction is actually known as Lohmann's reaction, and this actually serves as an immediate store of energy in the muscle whenever we require sudden energy. Therefore, creatine is a very important nutritional supplement for those who are trying to build body and muscle gains, right. So, we are coming back to synthesis of creatinine. So, what happens, we have got creatine phosphate which is actually spontaneously and non-enzymatically converted or broken down to creatinine, and this creatinine is excreted in urine. So, this reaction is pretty spontaneous and non-enzymatic, it is automatically converted to creatinine and an inorganic phosphate is let go from the creatine phosphate molecule. So, this is the highlight of the total synthesis of creatine. Mind it, these are the enzymes and these are the location where these enzymes are acting. So, all of them are again multiple choice question. The first step is in kidney, second step is in liver, and the third and fourth step are happening in the muscle cells. So, what is the clinical implication of creatinine? The normal serum level, okay, normal serum level is 0.7 to 1.4 in males. In females it is a bit less, 0.6 to 1.3 milligram per deciliter. Generally we measure creatinine level. Creatine level is not measured generally, even if it is measured, the normal level is 0.2 to 0.4 mg per dl. Similarly, in case of urine, creatinine as it is excreted in urine, it is the normal excretory product, so 1 to 2 gram per day, mind it is gram, whereas the value of creatine is 0 to 50 mg per day, it is almost negligible compared to creatinine excretion, right. So, all these levels are important and MCQ answers, right. So, what is the clinical implication of creatinine? It is a very important indicator of renal function, and the amount of creatinine actually parallels with the disease severity. The more damage the kidney, the higher will be the creatinine level in the serum blood, right. It is an indicator of glomerular filtration rate. Specifically, the 24-hour creatinine clearance, creatine excretion is very important indicator of, it reflects renal function. However, even if from a single value of creatinine, we can estimate the GFR roughly by using calculator. One such example is MDRD calculator. There are online softwares, you can simply put your age, sex, race and the creatine value, and it will give you a rough idea about the estimated GFR in your renal condition. You may like to try it, right. Next, this value is increased in muscular dystrophies, alright, both blood, urinary creatine and creatinine are raised. It is also raised in renal failure, fever and starvation, where there are, remember in starvation there is muscle degradation, in fever due to shivering, again there is a micro injury to muscle. Now, this enzyme creatine kinase is also very interesting, and it is a diagnostic enzyme because this enzyme, the level of creatine kinase, since it is present in inside cells, whenever there are destruction of the cells, this enzyme comes out, and its level is also an indicator of certain diseases. It is raised in myocardial infarction, muscular dystrophy, etc. And specifically you should briefly know, because this is not an enzymology class, but still you should know in this connection that there are 3 isoforms of this enzyme. The main isoenzyme that is increased in muscular dystrophy is CKMM variety, and when in case of heart disease, that is myocardial infarction, CKMB variety is raised, and hence it is an important marker of, it is an important cardiac marker, very early marker of and specific marker of heart disease, right. Moving forward, this creatinine production is actually very spontaneous, there is no fluctuation, and it actually depends on muscle mass. Therefore, creatine excretion is absolutely constant for a particular person, it will not vary, right. So, generally how the 24-hour creatinine is expressed is generally expressed as a amount of per gram, I told you per gram, and this creatinine is actually, it is so constant, it is also expressed, or it is used to normalize other parameters, for example albumin, they are expressed in mg per gram concentration of creatinine. So, it is also used to normalize the excretion of multiple other excretory products like albumin. So, albumin creatinine ratio is a very important indicator of renal function. So, as you have understood by now, creatine synthesis demands the major supply of glycine, arginine and methionine, told you MCQ. So, these 3 amino acids are very essential, and where there is an error in creatinine synthesis or this pathway, it has been found that those children are presenting with multiple neurological symptoms. Hence, it has been hypothesized that creatinine plays a critical role in brain development and brain function. Okay, it is still in research phases, but mind it, creatinine synthesis difficulty or problem leads to neurological symptoms in babies. So, next we moved on to another important product, that is heme synthesis. You all know heme is required for hemoglobin that is present in our blood, red blood cell, and this succinyl coenzyme A combines with glycine by the action of the enzyme delta aminolevulinic acid synthase. It forms delta aminolevulinic acid, which is also known as ALA, and this is the first step in production of heme. We will be discussing heme metabolism in across multiple classes in details for how, what are the steps, what are the diseases. So, for now you just need to know glycine is an important precursor for heme synthesis, alright. Next, purine synthesis, very important, interesting. Glycine, the whole molecule of glycine is utilized for synthesis of purine. Specifically, if we number the purine ring, we can see the 4th, 5th carbon and the 7th nitrogen are actually contributed by glycine. Again, we will be discussing synthesis of purine and nucleotide metabolism in detail in future classes. So, for now you should know that glycine is also acting as a precursor in production of purine nucleotides, right. Next, we move on to synthesis of glutathione. Glutathione is a tripeptide. It contains glutamic acid, cysteine and glycine. We have discussed glutathione when we were discussing about HMP shunt. We have discussed in detail how the reduced form of glutathione helps to fight various free radicals and how it helps in maintaining RBC membrane integrity, and this glutathione needs glycine to be synthesized, alright. So, there it happens in multiple steps, where first glutamate and glycine or glutamic acid and glycine forms gamma glutamyl with the help of the enzyme gamma glutamyl cysteine synthetase from gamma glutamyl cysteine, and then with the help of glutathione synthetase, since both require ATP, those are synthetase enzymes, it forms glutathione, which is also known as gamma glutamyl cysteanyl glycine. This whole phenomenon will also be touched upon again during absorption and digestion of protein in Meister cycle. So, be mindful, right, you have already read. Next, conjugation reactions. In conjugation reaction, glycine is an important conjugating agent for two things we will be discussing over here. Number one is bile acid. In cholesterol metabolism, you have been taught how cholesterol, with the help of several steps, several intermediate steps, they are converted to primary bile acids like cholic acid and chenodeoxycholic acid. They conjugate with amino acid like taurine and glycine. In this case, since it is a glycine class, we are discussing glycine conjugation. So, they are conjugated with glycine to form glycocholic acid and glycochenodeoxycholic acid. They can then form sodium, react with alkalis like sodium and potassium in the system, or cations, to form sodium glycocholate. So, that is how it forms bile salt, and thus glycine actually helps in synthesis of bile salts. So, this is conjugation of bile acid and formation of bile salt. Next, conjugation with benzoyl coenzyme. We have already read this, we are already familiar with this diagram when we discussed about ammonia metabolism, right, ammonia metabolism treatment. So, over there we discussed how this benzoyl CoA is actually conjugating with glycine to form hippuric acid, and thus it is excreted in urine, it is soluble, right. Benzoic acid is actually a preservative in food. So, what is the benefit of this? This is actually removing glycine from the system, and since glycine is an amino acid which needs to be synthesized in our system, subsequent synthesis of glycine removes excess ammonia from the system. So, this is an example of ammonia clearance with the help of conjugation that has already been discussed in detail in ammonia metabolism, alright. So, how glycine acts as a neurotransmitter? You see, glycine is actually present, the amino acid is actually present in brainstem and spinal cord, and it opens up specific chloride channel. Again an MCQ, multiple ion may be given, but glycine opens up chloride channel, and it in moderate level inhibits, it acts as an inhibitory neurotransmitter, but in high level it can cause overexcitation. So, this is a very careful balance of glycine that needs to be maintained in the tissue level, specifically at the central nervous system. And how glycine acts as a constituent of protein? Generally, glycine is present where polypeptide chain bends or turns. That is, when we look at the secondary structure, we have seen that whenever beta bends or loops are present, glycine is there because it helps in easy bending of the protein primary structure. I already told you it is a very important structural protein of collagen, and in collagen every third amino acid is glycine, again an MCQ, alright. Another amino acid is of collagen is proline, hydroxyproline, lysine, hydroxylysine, but the glycine is most common, alright. So, what are the metabolic disorders of glycine? So, I told you, remember the slides because that will be required when we are discussing disorder. So, glycine cleavage system, glycine was being broken down, is the most common breakdown pathway of glycine. So, if it is defective, what happens? Non-ketotic hyperglycinemia. Simple, glycine level will be extremely raised, it will lead to severe mental retardation and seizure, and there is no effective management. So, high level of glycine in blood, urine and CSF, glycine cleavage system problem. There is another disorder, glycinuria, which is basically a rare disorder, and in this what happens? Serum level of glycine concentration may be normal or decreased, but a very high amount is present in urine, because normally glycine is reabsorbed, but here glycine is not reabsorbed due to some problem in renal reabsorption, and hence it is characterized by excess glycine in urine, which is glycinuria, and then these patients have got an increased tendency to form oxalate renal stones. Okay. We also remember this slide where I told you this reaction where glycine is formed from glyoxalate strongly favors synthesis of glycine, but if there is some problem, there will be no formation of glycine and glyoxalate will be accumulated in excess, right, and this is leading to a disease that is known as primary hyperoxaluria. Excess amount of oxalic acid is excreted in urine and it is leading to the formation of oxalate stone. So, what happens? Basically, this is a protein targeting disorder where the enzyme that is normally present, it is supposed to be present in peroxisome. Due to a protein targeting defect, the enzyme is present in mitochondria. So, basically it is inactive in patients. So, if it is enzyme is inactive, this enzyme aminotransferase. So, what happens? This reaction cannot occur. So, glyoxylate cannot be converted to glycine, right. Moreover, in animals some glycine is actually oxidized spontaneously to glyoxylate. So, all of this leads to conversion of this glyoxylate to oxalate by the enzyme glyoxylate oxidase. GO stands for glyoxylate oxidase or glycolate oxidase, same thing. So, this leads to formation of excess oxalate, and thus excess amount of oxalic acid or oxalate are excreted in urine, right. There is another variety that is known as so what happens? So, let us discuss first. So, parts of oxalic acid, they could be directly oxidized to oxalic acid by glyoxylate oxidase. This is what I was discussing, alright. So, ultimately it leads to nephrolithiasis, that is formation of renal stones, renal colic, because once it is stone that will lead to excess pain, that is also known as renal colicky pain, and blood in urine, that is also known as hematuria, right. It so much so because all the organs are affected, oxalate crystal may be present in heart, blood vessel and bones, very serious disorder. There is a milder variety that is known as type 2 primary hyperoxaluria. Over what happens? The enzymes, that is glyoxylate reductase or hydroxypyruvate reductase, are deficient. Those are enzymes, because the glyoxylate normally, when it is formed in peroxisome, or it is normally channeled between the organelle and the cytoplasm. So, if the enzyme in peroxisome is fine, mainly I told you the reaction favors glycine synthesis, though the oxalate will be pulled from here. But even if some amount of glyoxylate is remaining in the cytoplasm, the normal pathway by which it was degraded, because normally it is reduced to form glycolate, then it is, all these things are basically hampered. So, what happens? This leads to production of oxalate, but I told you if this is only the reason, it is very mild, right. So, hydroxypyruvate deficiency, what happens? It leads to excess formation of glycolate and then glyoxylate to oxalate, right. So, this is a very milder condition and only leads to urolithiasis. The condition leads to production of stones only inside the kidneys, not extrarenal lithiasis. The primary one leads to production of stones in heart, blood vessel and bone. In this case, it is present, stones are only present inside the kidney. So, what is the treatment of this hyperoxaluria? Simple, we need to excrete oxalate by excess water intake. We need to take more and more water so that it is dissolved in water and excreted in urine. Dietary supplementation of calcium, it forms calcium oxalate crystals, and this will also help in chelation of the oxalate crystal excreted in urine, and dietary restriction of excess leafy vegetable like tea, beetroot, because they are a very important source of oxalic acid normally. So, if there is already oxaluria, we should keep in mind that these compounds which are a precursor of oxalic acid are restricted in diet. So, finally, we come to the slide where we are revisiting our original slides. We have discussed production from serine, threonine as well as 1 carbon, right. Proteins are broken down to form glycine by multiple proteolytic enzyme, that we all know, and finally, how glycine is utilized from, also that is important, glyoxylate. Glyoxylate also forms glycine, right. So, glycine cleavage system we have discussed, serine production of pyruvate to glucose we have discussed, glyoxylic acid to oxalic we have discussed in detail, creatine phosphate we will discuss in detail, production of purine ring we are discussing, purine metabolism we will be discussing, we have touched on, but we will still discuss heme synthesis in heme metabolism, we have discussed production of glutathione, we have discussed how it is conjugated in bile salt and conjugation of hippuric acid. So, this is the final metabolic fate that I always tell you, this is shown at the end of every amino acid metabolism class with a goal that our final amino acid, be it glucogenic and ketogenic, we are expressing the fate of carbon skeleton of amino acids. So, our player glycine lies here and it is converted to pyruvate and it being a glucogenic amino acid. So, to conclude, this lecture has covered the chemistry of glycine, how glycine is synthesized, we have discussed how glycine is degraded, we have discussed what are the important products from glycine, and we have discussed what are the disorders of glycine metabolism. So, this is the these are my few references for this slide, and I thank you all for your patient listening, and I see you in the next class. Take care.